Welcome back, Brain Bingers. I am so thrilled you decided to join us for a journey into the absolutely wild, slightly terrifying, and completely factual side of nature.
It's one of those topics that feels like it shouldn't be real.
It really does. We're going way off the deep end today, heading into the jungle floor where, true biological horror movies play out every single day. We're going on a brain binge into a topic that feels like science fiction, but is pure, highly specialized science.
And we are not talking about fiction, even though this phenomenon has inspired, video games and these huge TV shows.
Oh, absolutely.
The real science of how this works is arguably much more fascinating and frankly chilling than any scripted drama. This is the definition of highly specialized, high-stakes evolution.
It really is. So our mission today is to unpack the reality of the zombie ant phenomenon. We've been pouring through sources detailing the life cycle of one of nature's most effective puppet masters, the parasitic fungus, Ophiocordyceps unilateralis.
And its preferred victim, the carpenter ant, genus Camponotus.
Exactly. We want to give you all the specific scientific nuggets, the measurements, the chemistry, the mechanisms that truly define this system.
It's crucial to understand up front that this is not a random pathogen where an ant simply gets sick and dies.
Right. It's not like getting the flu.
Not at all. This is targeted, surgical behavioral control. The fungus doesn't merely seek to kill the ant. It needs the ant to perform a complex, choreographed series of actions at specific times in a specific microclimate to successfully complete the fungus's own reproductive cycle. It is, to put it mildly, biological precision at its finest.
And the initial hook, the aha moment that I think shattered assumptions in the scientific community, and it's where we need to start, is this. When you or I think of behavioral control, we automatically assume it must involve the brain, right?
Of course. Command center.
A command center,
Yeah.
like some chemical agent overriding the central nervous system. But the most surprising scientific fact about this entire ordeal is that modern imaging confirms the fungus actually avoids the ant's brain entirely.
That's the core breakthrough. For decades, the assumption was, well, there must be some kind of highly evolved neurotoxin. But the evidence now shows a complete body takeover, a physical and chemical hijacking of the muscle function.
A body takeover.
Yes. This allows the ant to remain mobile enough to carry out its grim instructions, but completely removes its free will. We're going to get into the specific mechanics of how it achieves that, because understanding how the puppet strings connect to the muscles, not the mind, that's the key.
Okay, let's unpack this biological masterpiece of horror, starting with the players involved.
Let's do it.
So let's start with the name. What exactly is a zombie ant? Because, even though we use the term, it is slightly sensationalized, but it's also accurate enough to describe the behavior, isn't it?
It is entirely descriptive, yes.
Right.
A zombie ant is an ant that has been infected and, crucially, controlled by a highly specialized parasitic fungus. The main culprit we focus on, the master manipulator, is Ophiocordyceps unilateralis.
And it's this control that's the key part.
It is. The fungus ensures the ant acts bizarrely. It loses its ability to follow the colony trails, it climbs vegetation, and most importantly, it dies in a specific locked position. They call it the death grip.
That locked position is the entire evolutionary point. The fungus isn't doing this randomly, is it?
Not at all. The ant's ultimate death position is entirely geared toward maximizing spore dispersal. It turns the ant into an unwilling mobile reproductive delivery system.
A living launch pad.
A living launch pad for the next generation of spores. Precisely. Now, if we look closer at the parasite itself, Ophiocordyceps unilateralis, we define it as an insect pathogenic fungus.
Okay, what does that mean exactly, insect pathogenic?
It's a vital classification because it means it has evolved for millions of years specifically to infect insects, and critically, its chemistry and anatomy are not adapted for mammal systems. Our sources are very clear. This is specialized to the insect world.
So that's a crucial distinction that separates the science from the scary movies.
A very important one, yes.
And its habitat is highly instructive about its life cycle needs, right? It thrives in tropical and warm temperate forests.
Mm-hmm. And when you picture that environment, the constant high heat, the dense undergrowth, the perpetually high humidity, you realize the fungus is adapted to a very specific set of environmental parameters. This dictates its entire strategy.
Including the specific death spot it programs the ant to find.
Exactly. And the specialization goes even deeper than just the climate. This fungus is what we call highly host-specific. Its primary victims are ants belonging to the Campanatini tribe, which includes the large carpenter ants.
So it's not just any ant.
No. Researchers have tried to infect related ant species, and while infection can occur, the behavioral manipulation is, well, it's significantly less effective. It gets sloppy.
That less effective manipulation is the telling detail, isn't it? That proves the specialization.
It is. It tells us that the fungus and the carpenter ant have been locked in an intense evolutionary arms race for a very, very long time.
Millions of years, probably.
Millennia upon millennia. The fungus has essentially tuned its chemical signals, its penetration enzymes, and its physical mechanics to perfectly match the neural and muscular biology of the carpenter ant. Any deviation from that host, and the hijacking becomes, well, non-functional.
So we're talking about a parasite that can only operate efficiently if it finds the exact right species of ant in the exact right humidity and the exact right temperature.
It's the ultimate specialist.
Okay, so how does this invasion even start?
Yeah. Let's move to how this complex invasion begins. For such a precise internal takeover to occur, the fungus first have to gain access, which is a surprisingly common ground-level process.
It starts simply, I assume, like ants are just walking around?
That's it. Ants, especially foraging ants, are constantly on the forest floor, on low vegetation, or along established colony trails. That's where they come into contact with the fungal spores that have been released from previously infected deceased ants.
So the spores are just waiting there.
They're everywhere. The fungus is successfully reproduced. And they're sticky. They stick immediately to the ant's outer shell, or its cuticle.
And once it's on, that's it. Game over.
Well, once attached, the clock starts ticking. But this isn't an acute infection. It's not fast. Our sources indicate that this infection process, from spore adhesion to internal proliferation, takes days to weeks.
Days to weeks? That's a long time.
It is, and this lengthy timeline is crucial because it emphasizes that this is a patient, highly strategic operation. It's a slow buildup of fungal biomass and chemical influence within the ant system.
Which allows the ant to keep acting normal for a while.
Exactly. It can remain mobile for the crucial behavioral modification stage later.
So the spore is stuck to the outside. Now here's the physical challenge I can't get my head around. How does something soft and microscopic, like a fungal spore, get past the hard, chitinous armor of a carpenter ant? That shell is designed for protection.
This is where the fungal assault becomes a masterpiece of biochemical engineering. The spores germinate and start producing these specialized structures called hyphae, which are essentially fungal filaments.
Like tiny roots.
Sort of, yeah. And these hyphae use a destructive combination of mechanical pressure, literally pushing and drilling through the cuticle alongside a potent cocktail of chemical weaponry.
Okay, let's get into the chemical specifics. What kind of weapons are in this fungal arsenal?
Absolutely. The fungus secretes specific, highly effective enzymes designed to chemically degrade the ant's complex protective layers. The sources specifically mention three main classes: chitinases, proteases, and lipases.
Okay, a coordinated, multi-pronged attack. Let's break that down. Chitinases. That sounds like it targets chitin.
Exactly. Chitinases break down chitin, which is the primary, incredibly tough structural component of the exoskeleton. It's the ant's armor. Without chitinases, the fungus couldn't get through the hard shell, period.
But the shell isn't just chitin, is it?
No, it's a composite material. So you have proteases, which are secreted to break down the proteins embedded in that cuticle layer. Those proteins give the shell flexibility and added resilience.
So it's softening it up.
It's dissolving the matrix.
Yeah.
And then you have lipases, which attack the lipids, or fats. Those are essential components of the outermost waxy layer, which helps the ant stay waterproof and prevents it from drying out.
So the fungus needs all three to ensure a successful, clean breach of the armor.
It does. It's not just brute force. It's a microscopic enzymatic siege weapon. It analyzes the defense structure, chitin, protein, fat, and deploys customized biochemical tools to dissolve its way through.
That level of targeted biochemistry it's just extraordinary.
And once it's successfully penetrated that outer defense, the fungus changes form again. Inside the ant's body, it transitions into a yeast-like state.
Why a yeast-like state?
It's an incredible adaptation. This form, known as blastospores, allows it to rapidly multiply and spread throughout the ant's hemocoel, which is the insect equivalent of a bloodstream.
So it uses the circulatory system to become systemic. It goes everywhere.
Correct. It quickly infiltrates all of the surrounding tissues. This internal growth phase, establishing the fungal network and accumulating the necessary biochemical metabolites.
Yeah.
This is what sets the stage for the dramatic behavioral transformation. The fungus establishes its internal network, preparing to throw the switch.
And override the ant's survival instincts entirely.
Completely.
Okay, the fungus is inside, it's multiplying, and it's spreading its influence throughout the hemicle. Now we hit the dramatic phase, the programmed death sequence. This is the part that truly shows how evolved this parasite is. It's a masterpiece of choreography.
It really is. The first observable stage is what we call stage 1, disorientation and descent.
The ant starts acting.
Weird, very weird.
Huh.
Infected ants begin to suffer convulsions and disoriented movements. They literally can't navigate correctly. They can't follow the chemical trails of their colony anymore.
And this causes them to fall.
This involuntary loss of motor control causes them to fall out of the high canopy, which is their usual foraging habitat, down to the lower vegetation or the forest floor.
Now, this is a good point for a probe. Why is the fall necessary? I mean, if the fungus is so good at controlling the ant, why induce a clumsy fall instead of just, I don't know, guiding it gently down the trunk?
That's an excellent question. The answer lies in those environmental constraints we discussed.
The humidity.
The humidity and the temperature. The fungus, while an expert manipulator, is highly specific about its environmental needs. The canopy is usually too hot and dry for optimal fungal reproduction. The fungus must maneuver the ant to its preferred spot, which is the cool, damp microclimate of the lower canopy.
So the fall is a side effect of the initial chemical attack, but a useful one.
A necessary consequence, yes. It forces the ant out of the safe but environmentally unsuitable high-rise habitat of the nest.
That makes perfect sense. The fungus sacrifices the ant's immediate coordination to force it into the reproductive zone. And then comes stage 2, the precise climb. This is where it gets deliberate again.
That precision is genuinely chilling. The ant climbs a plant stem or a leaf, but it doesn't just stop randomly. It halts at an optimized height. This is where the research data gets incredibly specific.
This is the part I love.
The ideal stopping point is usually around 25 to 26 centimeters above the forest floor.
Wait, 25 to 26 centimeters, that's roughly 10 inches, and that is astonishingly specific. How did researchers even figure that out? Does the fungus carry a tiny fungal ruler?
It's what makes this research so compelling. They didn't just measure a few dead ants. They conducted extensive field studies using environmental probes. They tracked hundreds of infected ants in natural environments and correlated the final death position with microclimate data collected at various heights.
So they were essentially building a 3D map of humidity and temperature.
Exactly. And they found that the target zone wasn't necessarily a fixed height, but rather a fixed microclimate. The 25 to 26 centimeter height correlates perfectly with a humidity level of 94 to 95%.
Whoa.
This proves the fungus isn't measuring distance. It is measuring environmental conditions and translating them into a command. Stop climbing when the humidity and temperature reach this perfect threshold.
So the ant is essentially a highly accurate living thermometer and hygrometer.
Programmed to stop when it finds the perfect conditions for the fungus's final reproductive growth. The level of environmental calculation is almost impossible to comprehend.
It really is. And once that precise spot is reached, we move to stage 3, the death grip.
This is the final defining act of the zombie ant. The ant bites down hard on a leaf or a twig, often targeting the underside of a leaf vein.
Why a leaf vein specifically?
Because veins offer a high concentration of structural support. It's a stronger anchor point.
And we know this bite is permanent. The mandibles lock shut, held closed by the fungal takeover of the muscle, and the ant never lets go. This is the anchor.
It is the ultimate commitment device. And scientists can track this activity, even long after the ant's soft tissues have decayed, because the bite leaves characteristic physical evidence.
The dumbbell shapes.
Exactly. Dumbbell-shaped marks on the plant tissue. Researchers use these marks to map out where the infection cycles are most intense, providing crucial data on spore graveyards.
Dumbbell-shaped marks, 25 centimeters high, 95% humidity. These are the details that elevate this from just a creepy story to true biological knowledge.
But there's one more layer of complexity to this stage.
Oh, there's more.
Stage 4, the timing. Observations in tropical forests reveal that many infected ants perform this final biting act around solar noon.
Solar noon, the middle of the day. Why? Wouldn't a nighttime bite be safer when predators are less active?
It seems counterintuitive from the ant's perspective, right? But it makes perfect sense from the fungus's. This timing suggests that light and daily rhythms or circadian cues are integrated into the manipulated behavior.
So what's the theory?
One hypothesis is that the fungus needs the ant to die and anchor itself at the warmest, driest point of the day to ensure optimal internal preparation before the stalk begins to grow. That final sun exposure might be necessary to harden the fungal tissues before the humid night.
Or maybe it's tied to maximizing exposure for the next day's activity. If the ant dies at noon, the fungus has the rest of the day to secure its anchor, and the spore release begins perfectly the next day when most ants are foraging beneath it.
That's a strong theory as well. Regardless of the exact chemical trigger, it underscores that these are not just random behaviors. They are precise, choreographed maneuvers dictated entirely by the fungal genome. The fungus controls the location, the action, and the timing.
Advanced behavioral engineering.
No other way to describe it.
This brings us to the most scientifically groundbreaking part of the research, which directly challenges everything we think we know about parasitic control. The mechanism itself.
The how.
Right. We teased the central fact earlier. The fungus does not penetrate the ant's brain. For something to control behavior, we instinctively look for manipulation of the nervous system, but modern science shows the fungus bypasses the ant's CPU entirely. Let's unpack that revolutionary discovery.
The scientific consensus is ironclad, established through sophisticated 3D imaging techniques like serial block face scanning electron microscopy.
That's a mouthful.
It is. But what these methods do is allow researchers to reconstruct the internal structure of the infected ant in stunning microscopic detail. And what these images confirmed was that while fungal cells proliferate throughout the ant's head capsule, they form a sheath around the brain, but do not infiltrate the central nervous tissue itself.
So the brain is structurally intact, it's just trapped.
Exactly. That is incredible technical work to prove a negative. But it raises a massive question. If the brain is structurally intact, where exactly is the command and control happening? If the steering wheel is untouched, what is the fungus manipulating to execute that precise climb and bite?
It has to be something else.
It's manipulating the power source and the levers, the muscles. Fungal cells surround and infiltrate the ant's muscle fibers, forming a complex, interwoven 3D network throughout the ant's entire body.
So it's like a web.
A web is a great analogy. This network is dense in the legs, which are essential for the 25-centimeter climb, and critically, it is highly concentrated in the muscles that operate the mandibles.
For the death grip.
For the final death grip, yes.
Oh, so the fungus is turning the ant's body into a biological marionette, where it is pulling the strings attached to the limbs and the mouth directly, rather than influencing the brain that generates the commands.
That's it. That explains why the movements are functional but clumsy. It's overriding normal control, but still using the ant's biological machinery. Researchers hypothesize that the fungus controls the muscles through a highly complex dual approach. First, there's the physical element, the vast network of fungal hyphae physically wrapping around and weaving among the muscle fibers.
So it's physically restricting them.
Restricting their movement and elasticity. But the mechanical aspect can't explain the complex, timed contraction of the death grip alone, can it? That requires a chemical command.
Right, it can't just be squeezing them.
Absolutely not. The more important and complex element is the biochemical takeover. The fungus is releasing a cocktail of specialized chemicals or metabolites. These chemicals likely interface directly with the muscle cells, changing their function and, crucially, altering the gene expression within the ant's own cells.
So it's a biochemical lockout. The ant's brain might be screaming, "Release the leaf, go back to the colony!" But the fungal chemicals are flooding the muscle receptors and forcing them to stay contracted.
That is the leading scientific theory. It seems to actively interfere with the ant's ability to relax the mandibular muscles, creating that permanent lock, even after the ant dies.
And here's the strategic genius of this muscle-focused system.
Yeah.
The ant needs to stay alive and mobile enough to execute the complex sequence.
Yes, the falling, searching, climbing 25 centimeters, and finally executing the precisely timed biting maneuver. If the fungus penetrated and killed the brain early, the ant would simply drop and die on the forest floor, a place too damp and unsuitable for optimal spore dispersal.
It requires a living, operational host for the final movement. The ant is basically biological equipment that needs to be functional right up until the point of installation on the leaf.
The fungus keeps the lights on, but takes away the keys.
This level of biological relationship is so integrated, it leads us directly to one of the most important concepts in modern biology when studying parasites. The extended phenotype. Let's spend a moment on that because it's a powerful term.
It is. An extended phenotype, a concept popularized by Richard Dawkins, is when the parasite's genes cause changes in the host's behavior or body structure that solely benefit the parasite's reproductive success.
It's the parasite expressing its own genes through another body.
Exactly. It means the parasite is expressing its own genetic agenda, its own survival mandate, through the body of another organism.
So every step we just described, the frantic climbing, the precise height selection, the solar noon timing, the irreversible death grip, is not ant behavior gone wrong.
Not at all. It is the direct, physical manifestation of the fungus's successful genetic programming. The ant's body becomes the parasite's puppet.
That is the key takeaway. And there's molecular evidence for this, right?
Oh, yes. The evidence supports this molecularly. Studies comparing gene expression in healthy and infected ants have shown simultaneous changes in the genes of both the fungus and the ant during the manipulation phase. For instance, the fungal genes related to producing those muscle-altering metabolites spike dramatically right before the final bite.
That's the smoking gun confirming the coordination. The fungus is turning on specific genes to create the chemical cocktail, and those chemicals are altering genes in the ant's muscle cells, forcing it to execute the final command.
This is a highly coordinated, evolved strategy, a biological masterpiece, and it underscores why this is such an important system for research. It gives us a clear window into how parasites can achieve such complex behavioral manipulation without needing to directly destroy the central command center of the host.
Okay, so the ant is dead. It's anchored to a leaf 25 centimeters up at 95% humidity. The fungus has achieved its goal, and now the victory phase begins. The focus shifts entirely from control to reproduction.
Indeed. What happens inside the body immediately after the death grip is essential for securing the launch platform. Our sources detail how the fungus continues to grow rapidly and fortify the ant's body.
Fortify it.
It essentially uses the ant's cuticle as a scaffold, reinforcing the exoskeleton, turning the corpse into a solid, durable shell that can withstand the elements.
Yeah.
It's not just a soft, decaying corpse. It becomes fungal infrastructure.
Wow.
And crucially, the fungus sends out specialized mycelial roots, those fungal filaments, deep into the plant surface around the mandibles. This mycelial growth anchors the body firmly to the leaf,
Providing structural reinforcement.
right, ensuring the death grip holds fast against wind, rain, and gravity for weeks or even months.
That's amazing. The ant warps becomes fully integrated into the leaf it died on.
Then comes the gruesome visual climax, the growth of the fruiting body, or the stalk. We call it a stroma. This is a dark, often cylindrical stalk that grows robustly out of the ant, typically emerging from the back of the head or the neck region.
That's the classic macabre image everyone associates with the zombie ant, the fungus erupting from the back of the neck like a biological antenna.
And that antenna is positioned perfectly. Once the stalk matures, and because it is positioned ideally, thanks to the ant's precise placement, it begins the process of spore release. It showers new infectious spores onto the forest floor directly below.
Maximizing the likelihood that new foraging carpenter ants will pass through this danger zone.
Exactly. And because the fungus is so good at selecting that perfect microclimate, often many infected ants die in the same highly favorable spots. This creates a striking and disturbing visual detail. Dense graveyards. Researchers have observed areas where these dead, anchored ants accumulate, sometimes reaching alarming densities up to 20 to 30 dead ants per square meter.
20 to 30 per square meter. Think about that concentration. It's a biological minefield just waiting for the next cohort of unsuspecting carpenter ants to pass underneath.
It is immense evolutionary pressure on the ant colony.
Which brings us to the inevitable counterargument. Ants are incredibly successful social insects. They are not defenseless victims. There has to be a robust evolutionary arms race happening here. How do the ant colonies fight back?
They fight back using a highly sophisticated system known as social immunity.
Social immunity, so group survival.
Exactly. It refers to a suite of cooperative behaviors that reduce the transmission and spread of the pathogen throughout the population. It's collective defensive action.
So what are the primary defense mechanisms they use?
The first line of defense is meticulous hygiene. Healthy ants will groom themselves and each other relentlessly, a behavior called allo-grooming, to remove any fungal spores.
So they're cleaning each other.
Constantly. This is a preemptive strike. They interrupt the initial stage of infection before the spores have the time they need to germinate and penetrate the cuticle. This behavior is metabolically costly, but absolutely essential.
Constant communal cleaning is the colony's antiseptic. What happens if an ant is already showing symptoms? If the fungus is inside and the ant is starting to get disoriented?
That's the second, and arguably more dramatic, defense mechanism: quarantine and removal.
Forced quarantine and banishment.
Yes. Colony members are somehow able to recognize an ant exhibiting early signs of the disorientation, the pre-zombie symptoms, before the final death grip phase. They will proactively carry that infected ant away from the nest and often dump it far away.
They are sacrificing that individual to limit the spore dispersal near the main population. That requires incredible sensory intelligence. How do they know?
It's a great question. They seem to sense the fungal metabolites or the altered movements. There are trade-offs here too. Removing a colony member is a loss of labor, but failing to remove them guarantees massive spore contamination, which would doom many more ants.
And finally, you mentioned an architectural defense.
Yes. Certain species of carpenter ants, especially those living in areas with high Ophiocordyceps presence, build their nests high in the canopy.
Stay off the ground.
Exactly. To keep most of their workers off the dangerous spore-filled forest floor altogether. By raising their collective habitat, they minimize exposure to the lethal spore graveyards below. This entire dynamic demonstrates the constant, intense evolutionary pressure. It's a never-ending arms race.
We've covered a remarkable amount of dense, thrilling material today. Before we wrap up, we absolutely must address the elephant in the room. The sheer terror this fungus inspires in pop culture.
That's completely understandable. We see this phenomenon referenced constantly, most famously as the inspiration for apocalyptic scenarios in media like The Last of Us,
where a fungus is hypothesized to jump to humans and control their behavior. So let's bring it back to Earth for the listener. Can this zombie ant fungus, Ophiocordyceps unilateralis, infect us?
The answer, according to all scientific literature, is a resounding no.
Okay, good to know.
The fungus is highly specialized for insects, particularly the Camponotus carpenter ants. Its entire mechanism, its chemical cocktail, its penetration enzymes like titinase, remember?
Right, for the exoskeleton.
Exactly. And its methods for bypassing the brain are all adapted exclusively to insect biology. The physiological gap between an insect and the human body is simply too wide for this fungus to bridge.
And our body temperature is a factor too, right?
A major factor. We are endothermic, warm-blooded, which creates a huge thermal barrier. Most insect pathogenic fungi cannot survive the 98.6-degree ARF human core temperature. It's a closed system. It remains a fascinating, albeit creepy, natural insect phenomenon, not a threat to people.
We can appreciate the horror without fearing the science. So let's do a quick synthesis of the key takeaways from this brain binge. What should our listener hold on to?
The core concepts are the highly evolved parasitic control, the crucial distinction that the fungus achieves manipulation through muscle invasion and targeted chemical signaling, actively avoiding the brain.
That's the big one.
It is. Then the concept of the extended phenotype, where the ant's final bizarre behavior is an expression of fungal genes, and the stunning level of specialization demonstrated by the exact measurements of the death grip.
The 25 to 26 centimeters high, timed around solar noon to hit the 95% humidity sweet spot.
That precision. It's a perfectly calibrated biological machine that uses a living ant as its final tool. It's absolute proof that sometimes the real-world biology is far more complex and captivating than anything we can invent in fiction.
Thank you so much for joining us for this extensive brain binge into the microscopic world of biological warfare. We hope this has given you specific, memorable, and shocking knowledge.
It's truly a great example of how examining detailed biological relationships can reveal such complexity. But let me leave you with a final provocative thought, something for you to mull over.
Lay it on us.
If a fungus has evolved to use complex, precise biology, using specific gene expression, chemistry, and muscle control, to command an ant to perform specific behaviors at a specific height at a specific time of day, what does this tell us about the hidden levels of biological control we might be overlooking in less dramatic ecosystems? We assume autonomy, but what other species are following unseen biological marching orders, guided by parasites or microbes that we simply haven't evolved the tools to notice yet?
That is a fascinating and somewhat chilling thought. Are we all just one highly evolved, host-specific metabolite away from climbing a leaf? That's something for you to explore next time you're walking through a forest and look down at the ground.
Indeed. Go forth and seek knowledge.
For now, that's all the time we have for this brain binge. Thanks for taking this dive with us, and we'll catch you next time.
That's it for this episode of Brain Binge. If you enjoyed getting your mind twisted tonight, do me a favor, comment, like, and subscribe so you don't miss the next deep dive. Got a weird fact, mystery, or place you want an episode about? Drop it in the comments. I read as many as I can, and I'm always hunting for new ideas. Thanks for listening, and thanks for letting your brain binge with me for a while. Stay well, stay curious, and I'll see you in the next one.